DNA replication is fundamental to life. Every time a cell divides, it must copy its DNA. This ensures that each new cell has the correct genetic information. Without accurate DNA replication, cells could not function properly, leading to many problems, including diseases. Replication is essential for growth, repair, and reproduction. When you grow taller or heal from a cut, your cells are dividing and replicating their DNA. Even single-celled organisms rely on DNA replication to reproduce. DNA replication also allows organisms to pass genetic information to their offspring. This process is crucial for the inheritance of traits. It ensures that each new generation has the genetic blueprint they need to thrive. Understanding DNA replication helps us grasp fundamental biological processes. It also aids in scientific advancements such as genetic engineering and medical research. By learning about DNA replication, we can better appreciate the complexity and wonder of life. DNA, or deoxyribonucleic acid, carries genetic information. It is composed of two long strands forming a double helix. Each strand consists of repeating units called nucleotides. There are 4 types of nitrogenous bases: adenine, thymine, cytosine, and guanine. Adenine pairs with thymine and cytosine pairs with guanine. The sequence of these bases encodes genetic information. This sequence is critical during replication. The first step in DNA replication is unwinding the double helix. Imagine a zipper being unzipped. This process is essential to expose the 2 strands so they can be copied. The unwinding begins at specific locations called origins of replication. At these origins, proteins bind to the DNA and help it unwind. This area is rich in adenine-thymine pairs because they are easier to separate than cytosine-guanine pairs. The unwinding creates a Y-shaped structure called the replication fork. As the DNA unwinds, it creates tension further down the strand. Enzymes called topoisomerases help relieve this tension. They make small cuts in the DNA, allowing it to untwist and then rejoining it. The unwinding process is controlled and precise. It ensures that the strands separate without breaking or tangling. This careful management is crucial for accurate replication, as each strand serves as a template for building a new complementary strand. Helicase is the enzyme responsible for unwinding the DNA double helix. Think of it as the motor that drives the unzipping process. It binds to the origin of replication and moves along the DNA, breaking the hydrogen bonds between the bases. As helicase moves, it separates the 2 strands of DNA. This creates single-stranded templates needed for replication. Helicase works at high speed, ensuring that the replication process is efficient. Its action is vital for the entire replication mechanism. The activity of helicase is tightly regulated. Other proteins assist helicase and stabilize the unwound strands. These proteins prevent the strands from rejoining before they are copied. This coordinated effort ensures that replication proceeds smoothly. Helicase is like a demolition crew in the replication process. It clears the way for the construction crew, which consists of other enzymes that build new DNA strands. Without helicase, the replication machinery would be stuck at the starting line. Section 5, DNA polymerase, the builder enzyme. Once the DNA strands are separated, DNA polymerase steps in. This enzyme builds new DNA strands. It reads the template strand and adds complementary nucleotides. DNA polymerase ensures correct base pairing. It needs a primer to start. Primase synthesizes this primer. DNA polymerase adds nucleotides one by one. It matches adenine with thymine and cytosine with guanine. DNA polymerase also proofreads and corrects errors. Section 6. Leading strand synthesis, smooth sailing. The synthesis of the leading strand is straightforward. This strand is built continuously in the same direction as the replication fork unwinds. DNA polymerase can add nucleotides smoothly, following the template strand. The leading strand requires only one primer to start replication. Once DNA polymerase attaches to this primer, it can keep adding nucleotides without interruption. This continuous process makes leading strand synthesis efficient and fast. The leading strand synthesis is like writing a sentence without lifting your pen. DNA polymerase moves along the template, adding bases in a smooth, linear fashion. This simplicity contributes to the overall speed of replication. Despite its simplicity, the leading strand synthesis is crucial. It ensures that one half of the new DNA molecule is made quickly and accurately. This efficiency is essential for the cell to progress through division without delays. Section 7. Lagging strand synthesis, the puzzle pieces. The lagging strand synthesis is more complex. This strand is built in the opposite direction of the replication fork movement. DNA polymerase can only add nucleotides in short segments called Okazaki fragments. These fragments are later joined together. Each Okazaki fragment starts with a primer. Primase adds these primers at intervals along the lagging strand template. DNA polymerase then adds nucleotides to form the fragment. Once a fragment is complete, a new primer is added further down the strand. The lagging strand synthesis is like writing a sentence in pieces, lifting your pen between each segment. This discontinuous process requires additional steps to join the fragments. As a result, it is more complex and slower than leading strand synthesis. Despite the complexity, the lagging strand synthesis is essential. It ensures that the entire DNA molecule is replicated. The coordination between leading and lagging strand synthesis ensures that both strands of DNA are accurately copied. Section 8. The role of Okazaki fragments, connecting the dots. Okazaki fragments are short DNA sequences synthesized on the lagging strand. They are named after the scientists who discovered them. These fragments allow DNA polymerase to replicate the lagging strand in manageable sections. Once an Okazaki fragment is synthesized, it must be joined to the previous fragment. DNA ligase is the enzyme responsible for this task. It seals the gaps between fragments, creating a continuous DNA strand. The process of joining Okazaki fragments is like assembling a jigsaw puzzle. Each piece must fit perfectly to complete the picture. DNA ligase ensures that the fragments are connected accurately, maintaining the integrity of the DNA molecule. Okazaki fragments highlight the complexity and precision of DNA replication. They demonstrate how the cell overcomes challenges to ensure complete and accurate replication. This intricate process is crucial for maintaining genetic information. Section 9. Conclusion. The precision of DNA replication. DNA replication is a marvel of biological engineering. It ensures that genetic information is accurately copied and passed on to new cells. The process involves many steps and enzymes, each playing a critical role in maintaining genetic integrity. The precision of DNA replication is essential for life. It allows organisms to grow, repair damaged tissues, and reproduce. Without accurate replication, cells could not function properly, leading to genetic disorders and diseases. Understanding DNA replication provides insights into fundamental biological processes. It also opens doors to scientific advancements such as genetic engineering and medical research. By appreciating the complexity of DNA replication, we can better understand the intricacies of life. DNA replication is a testament to the wonder of biology. It highlights the incredible precision and coordination required to sustain life. As we continue to explore and understand DNA replication, we unlock new possibilities for science and medicine.